So last time we were talking about enzymes needing coenzymes and cofactors to be fully functional. We said just the protein portion of the enzyme is the apoenzyme, and then its cofactor is an inorganic compound like a metal, like iron, zinc, or manganese. And then an organic cofactor, those are derived from vitamins, like the B vitamins especially. And once the apoenzyme has its cofactor and or coenzyme, it's complete, it's whole. So we call it the holoenzyme. And then it's functional. Now, enzymes can be inhibited. And there are two types of inhibition, competitive and non-competitive. In the case of competitive inhibition, there are substances that can bind to the active site of an enzyme and compete with the substrate for binding. So the idea here is that if the active site is always occupied by your competitive inhibitor, then the correct substrate cannot bind, which means the correct product cannot be made. So competitive inhibition is a concept that we humans have learned to exploit. We can use this natural concept to treat bacterial infections, for example. There are certain critical metabolic pathways that bacteria have that humans do not. And by finding drugs like antibiotics, which can bind to the active site of an enzyme and block it, that will prevent those bacteria from making that essential product. So we do have naturally occurring drugs like the antibiotics that we use to treat bacterial infections by acting as a competitive inhibitor and competing with the substrate. So you use enough of the competitive inhibitor so the active sites of the enzymes are always full of inhibitor, and that way the substrate cannot bind, and so the product cannot be made. And this must be a critical metabolic pathway, so that product needs to be a critical product that those bacteria must make. And so by blocking that enzyme, we shut down that pathway. And so those bacteria cannot make that essential product, and so they die. Certain toxins and poisons also work this way by being a competitive inhibitor. During the winter time, you always hear stories of people who die from carbon monoxide poisoning. And for whatever reason, either they're trying to use a gas stove, instead of to cook on, to use it for heat, which you should never do, or they have a backup generator, which is in the basement or garage, or it's too close to the house, so the fumes are coming inside. And the carbon monoxide competes with the oxygen for binding to hemoglobin. Only carbon monoxide has a much stronger affinity for hemoglobin than oxygen does. And so the carbon monoxide outcompetes the oxygen for binding to hemoglobin. And so you asphyxiate, you suffocate, you can't get enough oxygen because there's so much carbon monoxide bound up to your hemoglobin. So that's another example of a competitive inhibitor. Non-competitive inhibitors do not compete with the substrate for binding to the active site. What happens in non-competitive inhibition is there's an alternate binding site on the enzyme called the allosteric site. And when the non-competitive inhibitor binds to the allosteric site, it changes the shape of the enzyme, which changes the shape of the active site, so it can no longer bind to its substrate. So the allosteric inhibitor binds to an alternate site, the allosteric site, that changes the shape of the enzyme and the shape of the active site, so the enzyme can no longer bind to its substrate. This again is a process that humans can exploit to help treat certain infections, if both bacterial and viral. But non-competitive inhibition is actually a normal cellular process that cells use to purposely turn off an enzyme. So we'll talk about feedback inhibition in a moment. But non-competitive inhibition is a natural cellular process. Cells actually do this all the time to shut down an enzyme and shut down a metabolic pathway. So here we see our enzyme. We have the active site, and the substrate binds to the active site. And the old analogy they used to use is the lock and key, right? The lock is the enzyme, and the substrate is the key, so only one key fits that lock. It was never a great analogy if you knew anything about locks and how they work, but the idea is the substrate and the active sites are specific. So only one substrate can bind to the active site of a particular enzyme. Notice here we have that alternate binding site, the allosteric site. That is where a non-competitive inhibitor can bind and change the shape of the enzyme and the shape of the active site so the substrate can no longer bind. So this enzyme has been turned off. In the case of cells in a normal cellular process, the binding of the non-competitive inhibitor is reversible. So it's not permanently bound to the allosteric site. Right? The inhibitor comes on, it comes off, on and off, and it's concentration dependent. In the case of a competitive inhibitor, we're using some other substance like a drug which can bind to the active site and prevent the substrate from binding. In this case, you normally have to use a high enough concentration of that drug for a long enough period of time so the enzyme's active site is always blocked. And so cells cannot make that critical product and they die. So metabolic pathways are highly regulated. And this is important when it comes to saving energy. Making enzymes requires a lot of energy just to make the enzymes. And then for most enzymes to function, they use ATP in order to function. So a metabolic pathway requires a lot of energy, especially an anabolic pathway, a biosynthetic pathway. So if a cell does not need to make a particular product, right, because it's made too much, or maybe it doesn't need it right now, it needs a way to turn off that pathway to stop making a product it does not need, and that saves energy. And there are several different ways cells can do this. Allosteric regulation, we just talked about one example where an inhibitor molecule, a non-competitive inhibitor, can bind to an allosteric site and turn the enzyme off. But allosterism is a process that can be used to actually turn an enzyme on or to help stimulate it to function better. So allosteric regulation works both to activate and to inhibit enzymes. So most of the allosterically regulated enzymes are made up of multiple subunits. What level of protein structure is that, where you have multiple subunits? Well, right, the quaternary level of structure. So each enzyme has an active and an inactive form. An activator molecule can bind to the enzyme, stabilize the active form, right, to help keep it active. An inhibitor will stabilize the inactive form of the enzyme. And that's our allosteric inhibitor, the non-competitive inhibitor we just looked at. So here we see a multi-subunit enzyme. It has both an active form and an inactive form. An activator molecule can bind to this to stabilize the active form, and so the enzymes are all active. Or an inhibitor molecule can bind to the inactive form and keep it inactive. So these are allosteric forms of regulation because the activator and the inhibitor molecules are binding at some other site on the enzyme not the active site, but some other site. Now, cooperativity is a form of allosteric regulation that can amplify enzyme activity. So when the substrate binds to one of the active sites, it can stabilize all the subunits that are there together and make them active as well. So here we see the inactive form of an enzyme with multiple subunits and multiple active sites. When the substrate binds to one of the active sites, that activates all the other active sites. So it's just another way that enzymes can be regulated, because when a cell makes an enzyme, the enzymes are not, permanent. They don't last forever. And that's another mechanism to regulate activity inside of a cell. Enzymes degrade, right? They break down from use. And so cells have to make more of those enzymes. And this is another mechanism of regulation, because if a cell made an enzyme and that enzyme basically lasted forever inside of that cell, well, the cell doesn't always need that enzyme. It doesn't always have to be there. And so cells make enzymes as they're needed, and those enzymes eventually degrade, and then the cell makes more. And that also helps to regulate the metabolic activity inside of a cell. So there are many, many mechanisms of regulation. As I mentioned, there are drugs that we can use as allosteric inhibitors. An example here are the caspases, which are involved in inappropriate inflammatory responses. So when it comes to inflammation, in general, inflammation is actually part of your immune defenses. Some types of inflammation are bad inflammation, like when you strain a muscle or a tendon or something like that, and you get a lot of inflammation there. That's a harmful kind of inflammation. But when you have an infection or an injury, inflammation results. And the purpose of inflammation is actually to bring more blood to that site of injury or infection. And in the blood is your immune system. And so the purpose of inflammation is to bring your immune system to where it's needed. But sometimes inflammation is inappropriate or it's excessive, and you don't want that to happen. And so the caspases help to manage your inflammatory response. And caspases exist in both an active form and an inactive form. The active form can be stabilized when substrate binds, or the inactive form can be stabilized by that allosteric inhibitor, that non-competitive inhibitor binding to the allosteric site, and stabilize the inactive form. So it's just an example of how this regulation can be used in a cell, to regulate an enzyme, to activate it, or keep it deactivated. Not all enzymes are allosteric enzymes, right? So some of them are, and they have multiple mechanisms of regulation. Other enzymes are simpler and don't have allosteric sites. This is just showing you a filled-in model to show you the active form, and then the inhibitor binding to keep it inactive. So you notice there's a change in the shape, the inactive form versus the active form, right? Because when the inhibitor binds to the allosteric site, it causes a conformational change in the shape of the enzyme, and that changes the active site so it can no longer bind to its substrate. So here we just see an example of that, where the active form and the inactive form, so there are some differences in the shape of the enzyme. Now, feedback inhibition is a mechanism of regulation in order to shut down a metabolic pathway and stop making an end product. And so Metabolic pathways usually involve multiple enzymes, and so the allosteric enzyme is usually early in the pathway, so that when that enzyme gets shut down, there's nothing for the other enzymes to do. And since these enzymes use ATP to perform their reactions, this saves the cell energy. So when a cell makes too much of a product, the end product, so the actual end product, is the inhibitor molecule. So the product made by that pathway acts like a non-competitive inhibitor. It binds to the allosteric site of an enzyme early in the pathway. It turns that enzyme off, and that shuts down the whole pathway. As I mentioned, the binding of the non-competitive inhibitor is reversible and it's concentration dependent. So as the cell consumes the product, as it uses it up, it no longer binds to the allosteric site, the enzyme goes back to its normal shape and starts to function again. So the pathway gets turned on and the cell makes more product. If the cell makes too much product, then that product will feed back to that enzyme, bind to the allosteric site and turn that enzyme off and shut down that pathway until the cell uses up the product. And then the pathway gets activated again. And so many enzymes are regulated this way because sometimes cells make a little bit too much product. They don't need it all, right then. But they need a way to shut down that pathway just temporarily until the cell uses up the excess product and then the pathway gets turned on again. So this helps to save energy. So here we see an example, a multi-enzyme metabolic pathway to make an end product. If the cell makes too much of this end product, it can feed back to an enzyme, usually very early in the pathway, bind to the allosteric site and change the shape. That way the enzyme early in the pathway gets turned off. And so there's nothing for the other enzymes to do. So they just hang around and wait. Once the cell starts using up the end product, it comes off the allosteric site because the binding is reversible and it's concentration dependent. So as the cell uses up the end product, the concentration goes down. It no longer binds to the allosteric site, so the enzyme goes back to its normal shape and starts to function again. And so the cell can make more of that end product. So many biosynthetic pathways are regulated this way. So this is one way for the cell to save energy. Another way that we'll learn much later is that the end product can actually feed back to the gene and help turn the gene off. That way the enzymes don't get made in the first place. And that saves a whole lot more energy because making enzymes requires a lot of ATP. And so certain cells have ways to turn the gene off so the enzymes don't get made in the first place. And that will save even more energy. But we won't get to that for a while. But any questions on feedback inhibition, non-competitive and competitive inhibition? In eukaryotes, there are some enzymes that are found in specific organelles like the mitochondria. So remember, mitochondria have their own chromosome, they have their own genes, they have their own ribosomes, so they can synthesize their own proteins and enzymes needed for cell respiration, which we're going to talk about next. So here we see an electron micrograph of a mitochondrion. Remember, that inner mitochondrial membrane is very highly folded to increase surface area for all the electron transport chains that we're probably not going to get to today, but we'll get to that tomorrow then. So that's it for enzymes. Any questions? Now we're going to talk about cell respiration. And cellular respiration involves multiple types in order to make ATP. We're going to mostly focus on aerobic cell respiration. And then later we'll talk a little bit about fermentation, which is an anaerobic process. But it's important that we understand how cells make ATP, because we've learned last time how important ATP is and what it does. And remember, the hydrolysis of ATP is a spontaneous reaction. So it's an exergonic reaction. If you couple the hydrolysis of ATP with an endergonic reaction, a non-spontaneous reaction, you can make that reaction happen spontaneously. So cells use ATP for all kinds of work inside of a cell. And so ATP is the only form of energy that a cell can use directly. So as we'll learn, we eat food for energy, right? You eat sugar for energy. You can get energy from fat, even from proteins. But the energy stored in those food molecules cannot be used directly by the cell. It must be converted into a usable form, which is ATP. So we're going to start learning how cells do that today. So we're going to see all these letters, ATP, NADH, FADH2. So lots of letters. So it's an alphabet soup today to keep track of. This is one of those lectures where you need to write out and learn all the parts and pieces, and then see how those parts and pieces fit together. So all the energy we need must come from outside sources. Animals eat plants, and then other animals eat the animals that eat the plants, and so on and so forth. And plants get their energy from the sun. So plants form the base of almost all food chains on Earth, or at least photosynthetic organisms do. We've already learned that energy flows into an ecosystem as sunlight and leaves as heat. So heat is almost always produced and released when energy is transformed from one form to another. And the same is true with cell respiration. When cells make ATP, they also make heat. That heat is useful for many organisms. When it comes to photosynthesis, it produces oxygen strictly as a byproduct, as we'll learn later this week. And organic molecules, it makes carbohydrates, which can then be used in cell respiration. So plants, they use sunlight in order to make sugars. And then at night, when there is no sun, they can use those sugars in aerobic cell respiration to make ATP, just like animals do. But animals, basically are limited to aerobic cell respiration. But plants do both. So plants have mitochondria and chloroplasts. Animals just have mitochondria. And so the cells are going to use the chemical energy stored in those organic molecules and food molecules in order to regenerate ATP. Because as we talked about last time, when that terminal phosphate group is removed, and that change in the state of energy between ATP and ADP is what cells actually harness to do work. The ADP, the adenosine diphosphate, is quickly regenerated back into ATP. If you have to burn the ADP, you're left with AMP, so the cell's going to die, right? It's got to always have some ATP in order to make more ATP. So the ADP is quickly regenerated, it's quickly phosphorylated back into ATP. So just summarizing, light energy, our chloroplasts use that light energy to make oxygen and sugars, and those sugars can be used in aerobic cell respiration in the mitochondria, right, to make ATP. And of course, heat also gets released, and that heat helps to increase the entropy of the universe. Right? It's always increasing. Now, there are several ways that cell respiration happens, but there are some commonalities between the different forms of cell respiration. The breakdown of these organic food molecules is an exergonic process, so it is a spontaneous process. Fermentation is an anaerobic way to make ATP. And our cells are capable of doing this in a very limited fashion, but mostly we see this in microorganisms, in bacteria and yeasts. But the point is, no oxygen is required for fermentation, but that also severely limits how much ATP can be made. You really do need oxygen to make the maximum amount of ATP. So that's aerobic cell respiration. There are many, many anaerobic forms of respiration besides fermentation. I'm not going to go into those. Even if you take microbiology, you're probably not going to learn much about those because there are many different, pathways used by different microorganisms to make ATP, without any oxygen. Because remember, the first forms of life to evolve were anaerobic because there was little to no oxygen in the early atmosphere. And so all the microorganisms used anaerobic forms of respiration. When photosynthesis first began to evolve, oxygen was produced as a byproduct. As that oxygen accumulated in the atmosphere, microorganisms evolved to be able to use that oxygen to make even more ATP. And with all that extra ATP, cells started to associate together and form the first primitive animals, because you need a lot of ATP in order to make a complex multicellular organism. So cell respiration includes both aerobic cell respiration and anaerobic respiration, but most of the time when we talk about cell respiration, we mean just aerobic cell respiration. But any type of respiration by a cell which produces ATP is cell respiration. But mostly we talk about aerobic cell respiration. And carbohydrates are the primary source of energy, right? That's going to be used to make ATP, but fats and proteins, they can also be used to make ATP. But the overall simplified equation of aerobic cell respiration is what you see here. C6H12O6, which is our formula for monosaccharides. Here specifically, we mean glucose. So the C6H12O6 requires oxygen in order to burn it. So you can think of cell respiration as burning fuel. When you burn, something like wood or paper or gasoline, you need oxygen to burn it, right? Nothing burns without oxygen. Same thing with cell respiration. You can't oxidize that glucose. You can't burn it unless you have oxygen. And when that glucose is completely ripped apart carbon by carbon, all those carbons are released as carbon dioxide. So C6 in the glucose, we got 6 molecules of carbon dioxide. Water is also produced as a metabolic byproduct. So when you oxidize or burn a food molecule like glucose, you also produce a little bit of water as a byproduct. And there are 2 forms of energy, ATP and heat. Heat is actually the greater of the 2. So more of that chemical potential energy in the glucose molecule is lost as heat, as is made into ATP. And it's roughly about 40% ATP and 60% heat. So this is an equation that you must memorize. So you have to know this equation. So you must memorize that. So C6H12O6 plus 6 molecules of oxygen yields 6 molecules of carbon dioxide plus 6 molecules of water and energy in the form of ATP and heat. Now this is the simplified equation. It's actually a little more complicated than this, but all you need to know is the simplified equation. That glucose molecule has a lot of chemical potential energy in it. As the cell rips those carbons apart, it breaks those covalent bonds, it changes the energy state, but it's going to harvest those electrons, save them, use them in the end process to make the majority of ATP. But that glucose is going to be ripped apart carbon by carbon and released as CO2. But when you rip off those carbons, you break those covalent bonds, and those electrons are going to be captured, and they're going to be used later to make a lot of ATP. But you must know this equation. So the transfer of electrons during chemical reactions releases energy stored in those organic molecules. And this released energy is ultimately going to be used to make ATP. So as we'll see, it is all about those electrons. But we have to talk about oxidation and reduction first, because when electrons are given from one molecule or atom to another, oxidation and reduction occur. And so oxidation is always coupled to reduction. So we call them redox reactions. It's easier to pronounce than ox-red reactions, but redox reactions. So when a molecular atom is oxidized, it loses electrons. So it's lost those electrons, they have to go somewhere. And the molecule or atom that picks up those electrons, that accepts those electrons, it gets reduced, which sounds strange, right? Because you're gaining electrons, but you're being reduced, right? So it's a chemical term. So oxidation involves the loss of electrons, and reduction involves the gain of electrons. This can alter the charge of molecules or atoms. It can also produce stable covalent bonds. We talked about ionic bonds, right? Where the sodium, it donates an electron to chlorine, and so the sodium has lost some negativity, so it becomes a positively charged ion. And the chloride ion now, it has accepted a negative electron, so it becomes negatively charged. And so when oxidation and reduction occur, it can cause those molecules or atoms to gain a negative charge, or by losing negativity, it ends up with a positive charge, or they could be partial charges. But oxidation and reduction are coupled together. And so what I just described with the sodium and the chlorine, a more general type of equation, an electron is donated from one atom or molecule to another one. The atom or molecule that loses the electron becomes oxidized, and the atom or molecule that gains the electron becomes reduced. There are two ways to remember oxidation and reduction. One of them is oil rig, which stands for oxidation is loss, reduction is gain, or Leo the lion goes grr. Lose electrons oxidation, gain electrons reduction. So two ways to remember what is oxidation and what is reduction. But whenever an electron is lost, it's got to be gained by something else. Now here's a concept that's a little confusing. The electron donor is called the reducing agent. So the electron donor will become oxidized because it's lost an electron, but it's giving that electron to another molecular atom, which becomes reduced. So the electron donor is called the reducing agent, even though the donor is becoming oxidized. It's reducing something else. And the electron receptor or acceptor is called the oxidizing agent. So the electron receptor becomes reduced when it gains the electron, but because it's gained it from something else, it caused that something else to become oxidized. And so the electron receptor is called the oxidizing agent. So again, the reducing agent has lost electrons and given them to something else. And the oxidizing agent has gained electrons, thereby causing something else to become oxidized. Any questions on that?
Professor, before you move on, you mentioned that one of them is covalent bond and one is ionic. I missed that. Can you please come again?
Well, no, it's just that when electrons are donated, sometimes it can produce a covalent bond or it can produce an ionic bond. It can result in charges on the molecules, or they can share those electrons.
Okay.
So during cell respiration, the fuel, which is most commonly glucose, is going to be oxidized, and the oxygen is going to be reduced. Right, so a glucose molecule is a fully reduced molecule, right? It's got all the electrons it needs, all the hydrogens it needs, it's fully reduced. Now the oxygen, which is O2, is bound to itself, but it can be further reduced. It can gain more electrons and become water. So here's our fully reduced molecule glucose. As it becomes oxidized, it turns into CO2 because it's losing electrons to become carbon dioxide. The oxygen is being reduced because it's gaining electrons to become water. So the glucose will be oxidized and the oxygen will be reduced. And we end up with CO2 and water, and in the process, energy is going to be formed. But the glucose, as it exists as a glucose, is a fully reduced molecule, right? It's loaded with electrons. When you rip it apart and break those covalent bonds, those electrons will go to the oxygen to form water. And the carbons end up being carbon dioxide. So again, this is our formula for aerobic cell respiration. But we see how the glucose is being oxidized to carbon dioxide, and the oxygen is being reduced into water. And again, this is the simplified equation. Now, NAD plus, right, it's charged because it's oxidized right now. It's going to accept electrons and a hydrogen to become reduced and form NADH. And the NADH is one of our very important electron carriers. So as we'll see, as the carbons are ripped apart from that glucose molecule and the covalent bonds are broken, electrons will be captured by NAD plus to make NADH. And there's another electron carrier we'll see later, FAD, which gets reduced to FADH2. But these are two extremely important electron carriers. And they're going to carry those electrons to the electron transport chain to make ATP. So the chemical potential energy stored in glucose is in the electrons, and the electrons are going to be captured in NADH as well as some FADH2. But those electrons are going to be used ultimately to make the majority of ATP in a cell. So NAD plus becomes NADH when it's reduced. FAD becomes FADH2 when it's reduced, but they're carrying electrons because they've been reduced. Now, for the biological reactions we're going to talk about, oxidation and reduction involves not only the transfer of electrons, but also hydrogens along with those electrons. So that's why we see reduced molecules like NADH, right? It's got electrons, but also a hydrogen. FADH2, it's got electrons, but also hydrogens. So for biological reactions where oxidation and reduction are involved, hydrogens are also transferred along with the electrons. So NAD is a coenzyme. It's also a very important electron carrier. And those electrons are going to be used to make ATP. So the glucose, all that chemical potential energy in those covalent bonds, those covalent bonds are going to be broken as the carbons are ripped apart one by one. And those electrons are going to be captured by NADH. Later on, we'll see some capture by the FADH2. But the NADH is carrying those electrons, and it will carry those electrons to the electron transport chain to make ATP. So lots of letters. It is an alphabet soup today, but hopefully by the end of tomorrow, it'll all come together. So as I mentioned, the electrons are going to be carried to the electron transport chain. And the electron transport chain is a controlled series of reactions which release energy slowly, step by step. So if you kind of think back to electrons in the orbitals and shells of an atom, the outermost, electrons had the highest energy. As they drop down to another shell, they lose energy, they have lower energy. And so when it comes to using these electrons and the stored energy in the electrons, it's going to be released in a stepwise fashion with little bitty amounts of energy actually released. And remember, some of that energy, in fact, most of it is heat. And so when you burn fuel, it produces heat. When a cell burns the glucose, when it oxidizes the glucose, it'll release heat, but you don't want that heat released all at once. It would be damaging to a cell. And so the heat's going to be released in little bitty steps. If too much energy gets released all at once, it can have really negative consequences. So here we see a way to illustrate how the energy stored in the electrons is going to be released in little steps in the process of electron transport. The hydrogens, along with the electrons, are going to be used to make ATP. The electrons serve as one alternate form of energy, and the accumulated hydrogen ions will serve as another form of alternate energy, which allows the cell to make ATP. But hydrogen, even though it's the simplest element, right? Only it's basically just a proton. A hydrogen molecule is H2, right? Two hydrogens bonded together. But in the presence of oxygen, it can be explosive, right? Hydrogen burns. Maybe you've heard of hydrogen cells, right? You use a hydrogen fuel source, so we can, make our cars move without burning hydrocarbon fuels like gasoline. Well, the hydrogen is potentially very, very explosive if the energy is released all at once. And a real life example where that happened was the Hindenburg, disaster. And this happened in 1937. The Hindenburg was a blimp or a Zeppelin, a German Zeppelin on its way from Germany to New Jersey. And when it got to New Jersey, something happened. A spark, from somewhere ignited the hydrogen because the blimp was filled with hydrogen, which is very explosive in the atmosphere. Helium, another very light gas that we use in balloons and things like that, is not flammable. It's not explosive. Germany had no natural source of helium, but it did have hydrogen, and that's what it used in its Zeppelins. So a big floating bomb, very highly explosive. And something happened when it got to New Jersey. And there was a spark that ignited all that hydrogen, and it exploded and burned. It's a very, very famous film. If you just go to YouTube or Google it, you'll find the film with that very famous line from the reporter that was there. Oh, the humanity, right? So that's where that line comes from, was the Hindenburg disaster and that reporter who was there on the scene. But it's an example of how hydrogen has a lot of tremendous potential energy stored in it. And when it's released all at once, it's explosive. And so the cell doesn't want to explode, right? It releases that energy in little bitty steps. It does generate heat, which is useful for many animals, but it's done in a very controlled way. So, but you should take a look at the Hindenburg disaster just to see what happened. But they were using hydrogen, very dangerous, extremely dangerous, because Germany had no helium, and so they had to use hydrogen. So, cell respiration, aerobic cell respiration, has 3 stages. And the first stage is glycolysis or glycolysis, right? Glyco refers to sugar, in this case glucose, and lysis means to split, right? To break it down. The citric acid cycle goes by different names. I prefer the Krebs cycle, named after the scientists that helped to figure it all out. But the citric acid cycle, it's used to also be called the tricarboxylic acid cycle, or just the Krebs cycle, but I like using Krebs, so the scientist gets the credit. And then we have oxidative phosphorylation, along with the electron transport chain. And that's where the majority of ATP gets made at the very end of the process. And this is a long process with many, many biochemical reactions. And all those reactions are required to make all this ATP, but there's a lot of enzymes involved. And there is some hydrolysis of ATP in order to start the process. And that's why you need some ATP in order to make more ATP. But glycolysis, Krebs, and the electron transport chain and oxidative phosphorylation. When you phosphorylate, you're adding a phosphate group onto a molecule. There are actually 3 ways for cells to phosphorylate to make ATP. Oxidative phosphorylation, as we'll see shortly, substrate level phosphorylation, where a phosphorylated organic compound transfers that phosphate group to ADP to make ATP. And then a photophosphorylation, which is the process of photosynthesis. And those are the 3 ways to phosphorylate, to add a phosphate group onto adenosine diphosphate to make adenosine triphosphate. Either oxidative phosphorylation, substrate phosphorylation, or photophosphorylation. And later this week, we'll get into photosynthesis. But these are the 3 stages of aerobic cell respiration: glycolysis, Krebs cycle, oxidative phosphorylation, and the electron transport chain. So glycolysis occurs in the cytoplasm of all cells. As we're going to see, our glucose molecule is going to be split into pyruvate, also known as pyruvic acid, which each contain 3 carbons, so there's 2 of them. So we're simply splitting our 6-carbon glucose into 2 3-carbon pyruvic acids, or pyruvates. In the process, a little bit of ATP is going to be made, and it's made by substrate level phosphorylation. So a phosphorylated organic compound is going to transfer that phosphate group to ADP to make ATP, but not much. NADH is going to be produced, and the NADHs are carrying electrons to the electron transport chain to make ATP. In a eukaryotic cell, we now move into the mitochondrion to continue with the next 2 stages. In prokaryotes, they have no mitochondria. So there are some differences between prokaryotes and eukaryotes when it comes to making ATP. But in the eukaryotic cell, our Krebs cycle, the citric acid cycle, occurs inside the mitochondrion. As we'll see, a little bit more ATP gets made by substrate level phosphorylation. A lot of NADH gets made. And remember that NADHs are carrying electrons to the electron transport chain to make ATP. Also, a little bit of FAH2 gets made. And they are also carrying electrons to the electron transport chain to make ATP. But what the cell is really doing here is it's oxidizing that glucose. It's ripping it apart carbon by carbon. It's breaking those covalent bonds, and it's capturing those electrons, mostly in NADH, a little bit of FADH2, but those electrons are going to be used to make the majority of ATP inside a cell. So the cell's not getting much ATP until the very end. It gets a little bit from glycolysis and Krebs, but most of it comes with electron transport chain and oxidative phosphorylation. A chemiosmosis, you know osmosis and chemi, so it's not water, it's actually hydrogen ions, which again, this process will happen inside the mitochondrion of eukaryotes. But we're going to learn how cells make ATP by substrate level phosphorylation just a little bit. And then we'll see how the cell makes a lot of ATP with oxidative phosphorylation. And as we'll see, oxidative phosphorylation requires oxygen. So we're going to see where the oxygen finally comes in. So oxidative phosphorylation involves a lot of oxidation and reduction. So a lot of redox reactions will occur. Oxidative phosphorylation makes most of the ATP in a cell. About 90% of the ATP made by aerobic cell respiration happens at the very end of the process with oxidative phosphorylation. And the other 10% comes from glycolysis and Krebs and substrate level phosphorylation. So it's not very much. So that's why we need the oxygen to make a lot of ATP. So here's to showing you substrate level phosphorylation, which does require an enzyme to make this happen, but you have an organic compound that's phosphorylated, and that phosphate group will be transferred to ADP to make ATP. And this does require an enzyme to make that happen. So all the steps we're going to see require many, many different enzymes. Fortunately for you, you do not need to know them. If you go on to take biochemistry at a 4-year school, you will have to learn all of them. So, as I mentioned, glycolysis or glycolysis is where glucose is going to be split. It produces 2 pyruvates or pyruvic acid molecules, each containing 3 carbons. So only one covalent bond that gets broken in glycolysis. All the rest happen in the Krebs cycle. So the cell needs to make ATP. If you think of ATP as money, how does that old expression go? What do you need in order to make money? What do you need to make money? How's that old expression go? Right, you need money to make money, right? You need to be able to invest money in order to make more money. It's the same thing with ATP. A cell has to invest some ATP in order to make more ATP. So the cell has to always have some ATP in order to make more. And so the ADP is going to be regenerated back into ATP very quickly. So the cell can keep on making more ATP. If cells are starved, they use up all the ATP, then they use up all the ADP, and then they die, right? Because they can't make any more energy, right? They've got to have some ATP to start the process. And that occurs in glycolysis. So you can think of ATP as the energy investment phase, or I like to think of it as a way to jump start the process of glycolysis. You know, like you jump start a dead battery, right? You need some energy to jump start it to get to go. So you need here some ATP to jump start glycolysis and get it to go. And then you have an energy payoff phase. So you will make more ATP than you invested. So ATP is used to start the process of glycolysis. By the end of glycolysis, there will be 2 molecules of pyruvate or pyruvic acid. 4 ATPs get made, but because the cell had to burn 2 ATPs to start the process, we always just talk about a net production of 2 ATPs from glycolysis. So yes, technically the cell will make 4 ATPs, but it had to burn 2 ATPs in order to make the 4. So the net production is 2. And that's all we say that glycolysis produces is 2 ATPs. It also has produced 2 NADHs. Those NADHs are carrying electrons to the electron transport chain to make ATP. So the cell's already started to save or store, right, electrons in order to make ATP. So the cell's made a little bit here, right, just 2 molecules of ATP in glycolysis. The pyruvates are molecules that each contain 3 carbons, right? So there's 2 pyruvates. They each contain 3 carbons. They have to enter into the Krebs cycle next so that each of those carbons can be broken and the covalent bonds broken and the electrons captured. But to go through the reactions, which you don't need to know, but I just want to show you all the different enzymes here. Hexokinase is one that uses ATP in order to phosphorylate our glucose. And then that glucose 6-phosphate is going to be converted into fructose 6-phosphate. You don't need to memorize these. But another enzyme, phosphoglucoisomerase, is what does that. So it converts the glucose 6-phosphate into fructose 6-phosphate. And then the fructose 6-phosphate is again going to be phosphorylase, so another ATP is going to be used here. And the enzyme is phosphofructokinase. Don't need to remember these enzymes. And then we have a double phosphorylated fructose molecule. And that fructose molecule that's phosphorylated is going to be converted into glyceraldehyde 3-phosphate or dihydroxyacetone phosphate. They're both isomers of each other. And the glyceraldehyde 3-phosphate is going to be converted into bisphosphoglycerate. And here is where the electrons get captured by the NAD+ into NADH. And since there's 2 glyceraldehyde 3-phosphates, this will happen twice, right, to make our 2 NADHs, right? Because we're splitting our glucose, which was a 6-carbon molecule, into 2 3-carbon molecules, and this is the intermediate, 1, 2, 3 carbons here, and there's 2 of them. And so this happens twice, so we'll make 2 NADHs. And then the bisphosphoglycerate, it's got those 2 phosphates. It's going to donate those phosphates to the ADP to make ATP. And again, this will happen twice because there's 2 of those molecules. And some more conversions. And we finally, again, more ATP is made. Remember, a total of 4 molecules gets made, but since the cell had to use 2 molecules to start glycolysis, we just talk about a net production of 2. And we finally end up with our pyruvate or pyruvic acid. And there's 2 of these, right? Because our 2 3-carbon pyruvates originally formed a 6-carbon glucose. We split that 6-carbon glucose in half. So now we have 2 3-carbon molecules. In the process, the cell made some ATP, a net production of 2 ATPs, but also 2 NADHs. And what are the NADHs carrying? What are the NADHs carrying?
Electrons.
Electrons. To where? Starts with electron.
Electron transport chain.
Right, the electron transport chain in order to make what? What's the cell trying to make?
ATP.
ATP, right? So it's all about the electrons, and those electrons come from the covalent bonds between the carbons, and those covalent bonds are going to be broken, and the electrons captured in NADH mostly, a little bit of FADH2 later, but all those electrons are going to be used in the electron transport chain to make ATP. So the 2 pyruvates, they cannot enter into the Krebs cycle directly. They must be converted into acetyl coenzyme A, also known as acetyl-CoA. And we call this a decarboxylation step because some carbon dioxide is now going to be released here. Also, some more NADH will be made in this intermediate step. If we're in a eukaryotic cell, this has now happened inside the mitochondrion. If we're in a prokaryotic cell, like a bacterial cell, we're still in the cytoplasm. But we're talking about eukaryotes here mostly. So we're inside the mitochondria now. That's where the decarboxylation step occurs. So our pyruvate, and remember there are two of these, so what you see will happen twice. It's going to be decarboxylated to produce acetyl coenzyme A. So one of the carbons gets ripped off here and released as carbon dioxide. Because a covalent bond has been broken, electrons have been released and captured by the NAD+ to make NADH. So remember, there are two pyruvates, so this happens twice. So we have 2 molecules of CO2 get released, 2 molecules of NADH are produced, and there are 2 molecules of acetyl coenzyme A, which can now enter into the Krebs cycle. Also known as the citric acid cycle, but I like Krebs. It happens within the mitochondrial matrix. So in the space between the inner and the, well, in the center of the mitochondrion, surrounded by the inner mitochondrial membrane, is the matrix. And so that's where the Krebs cycle takes place. And so as we're going to see, there are many different intermediates in the Krebs cycle. Again, you don't need to know what the intermediates are or any of the enzymes or reactions. What we like to focus on, or what I like to focus on, is what gets made, how much of it gets made, and where it gets made. So for each molecule of pyruvate, 1 molecule of ATP will be produced, 3 molecules of NADH, and 1 molecule of FADH2. So that's for each pyruvate. Remember, there are 2 pyruvates, which make 2 acetyl coenzyme A's. And so the overall Krebs cycle will yield 2 molecules of ATP, 6 molecules of NADH, and 2 molecules of FADH2. So here we see the Krebs cycle. We're in the mitochondrial matrix now. If we were in a prokaryotic cell, we would still be in the cytoplasm. But since we're in a eukaryotic cell, we're inside the mitochondrion. So our pyruvate gets decarboxylated to make the acetyl coenzyme A. We said that CO2 gets released and NADH is produced. Now the acetyl-CoA is going to cycle through the Krebs cycle, make NADH, ATP, and FADH2, and every carbon is going to be ripped off and released as CO2. And when those covalent bonds are broken, they'll be captured by the NADH and a little bit in the FADH2. So there are 8 steps, 8 specific enzymes, 8 intermediate molecules, none of which you need to know. Lucky for you. But the acetyl-CoA is going to be combined with a molecule called oxaloacetate, which will form citrate. And the next 7 steps help to break down that citrate into oxaloacetate. In the process, all those carbons are going to be released as CO2. And the NADH and FADH2 produced by the cycle are carrying those electrons to the electron transport chain to make ATP. So all the different intermediates, none of which you need to know. Lucky you. But acetyl-CoA combines with oxaloacetate, and that makes citrate, and then we go through all these different intermediates: isocitrate, alpha-ketoglutarate, acetyl-CoA, succinate, fumarate, malate, and back to oxaloacetate, which will combine with the next molecule of acetyl-CoA and continue the cycle. So because there are 2 acetyl-CoAs, because there were two pyruvates, the cycle you see here happens twice. So one NADH here, another NADH here, another one here, 3 NADHs per cycle. It happens twice, so that's 6 NADHs. Notice some ATP gets made. 1 ATP for every acetyl-CoA, so there's 2 acetyl-CoA, so 2 ATPs. And here we have an FADH2. And since this happens twice, there are 2 FADH2s. So a lot of NADH, a little bit of FADH2, and a little bit of ATP. You have a question, Crystal?
I do. I was trying to wait for you to finish. So, so because the Krebs cycle, it happens twice, it double up on the end result?
Yep.
Okay.
So let's see how well you guys have been paying attention. So what is the net production of ATPs so far? How many ATPs since the very beginning? We're talking about the overall net production has the cell made. So how many ATPs since the beginning? And just the net production of ATPs total since the beginning. How many were made in glycolysis? 2. Were any ATPs made in the intermediate step, the decarboxylation step? Nope. And how many ATPs are made in the Krebs cycle? In the Krebs cycle? 2, for a total of 4. So look at all the effort the cell has gone through. All those different intermediate molecules in glycolysis, all of those enzymes. And here we see a lot more intermediates, a lot more enzymes, a lot of work for 4 lousy stinking molecules of ATP. Boy, what a waste. That's a lot of effort for so little ATP, right? Just 4 lousy molecules of ATP? That's not much. But what has the cell made a lot of? NADH. How many NADHs since the beginning? Are you sure, Crystal? Remember, 2 NADHs in glycolysis, 2 in the intermediate step, and 6 in the Krebs cycle for 10. Plus 2 FADH2, so that's 12 electron carriers. And those carriers are going to carry those electrons to the electron transport chain to make ATP. So what the cell has put all this effort into doing is capturing and storing electrons. So basically storing potential energy, which is going to be used to make ATP. So a little bit of ATP has been made in glycolysis and Krebs, and that's the result of substrate level phosphorylation. So not much ATP, but a lot of electron carriers, especially NADH, have been made. And those electrons are going to be used in the process of electron transport in order to make the majority of ATP in a cell. And so all this effort is worth it, because as we're going to learn tomorrow, in order to make ATP, you need alternate forms of energy in order to make ATP. All right, so we need ATP to start the process of glycolysis, but when we get to the end, we need a way to make ATP without using ATP. Otherwise, there'd be no net gain. And so alternate forms of energy have to be created. One of those alternate forms of energy is electrons and in the electron transport chain. So we're gonna stop here for today because that's a lot. And so we'll finish up this tomorrow and talk about the electron transport chain and chemiosmosis and how the cell makes most of its ATP. We'll also look at fermentation as our only form of anaerobic respiration we're going to talk about. So I know it's a lot. It's an alphabet soup: ATP, NADH, FADH2, right? So, but you need to take the time to look at all the parts, look at all the pieces, write out what each piece does, and so you can help put it all together tomorrow. Two of the NADHs are made in glycolysis. Two were made in the intermediate step, the decarboxylation step, and then 6 are made in the Krebs cycle for a total of 10. And then we have those 2 FADH2s, which also donate electrons to the electron transport chain to make ATP.
So during glycolysis, does it make FADH too?
No, the FADH2 is only made in the Krebs cycle.
Okay, that's why I was confused.
And it's just a little bit, just 2 molecules. Most of the electrons come from the NADH and are used to make ATP. Okay, so like I said, we'll stop here for today. We'll finish this up tomorrow. And then